Hi, Assalamualaikum. My name is Siti Nuatira binti Khairi and my metric number is 196872. And on this video, I want to share with you about the enzyme link receptor. Enzyme link receptor, like all receptor, it receives signal from the environment and they instruct the cell to do certain thing. Enzyme link receptor are cell receptor associated with intracellular domain and with an enzyme, and in some cases the intracellular domain of the receptor itself is an enzyme. Binding of a signaling molecule activate the receptor enzymatic activity. Let's go further on the structure of the enzyme link receptor. The general structure consists of extracellular domain, which can bind a ligand or known as ligand binding domain. Down here on the intracellular side is the enzymatic domain. Enzyme link receptor that we identify so far are tyrosine kinase receptor, tyrosine kinase associate receptor, receptor like tyrosine phosphate, receptor serine phosphorylate, and histidine kinase associate receptor. Though there are many different types of enzyme link receptor, the most widely recognized and most common is called receptor tyrosine kinase. They are important because they regulate cell growth, differentiation, and survival. This is receptor tyrosine kinase. Tyrosine is an amino acid and right here is a polypeptide. Here is the signal binding site where the ligand will bind into it. When the ligand binds to the signal binding site, these two separate polypeptides are going to aggregate or come together, and here is called dimers. This dimers is unphosphorylate dimers, but it does have activated tyrosine. A kinase transfer phosphate group, so here the phosphate is taken off from ATP and attached to one of the tyrosine. So basically, the kinase activity taking phosphate from ATP and then phosphorylating these dimers. Now the dimers have become phosphorylated dimers. The next step is a relay protein, which separate protein coming in and take the phosphate away, and this protein will go through a conformational chain shape. It will lead to own signal transduction pathway and a cellular response. A different protein will come in and will be activated by different tyrosine region. And next, we'll activate a different signal transduction pathway and a different cell response. After all the phosphate is taken off, finally the ligand is dissociated from the binding site. The dimers break apart. For me, I think this STEM teaching tools is very helpful and effective for learning process because we can see more clearly on how the process going on. And these STEM teaching tools enable our generation to innovate. Students are able to design, build, and control a variety of tools to make it more fun learning. Innovation leads to new products which help learning process to become better and to have a better understanding. Thank you.